help me to just be clear and concise, the things that are in my head and my heart to be able to teach about this life that you've given us and your good plan for life continuing on and multiplying with relation to DNA and RNA and proteins. Father, may it just be very useful to all of us. Help us to have minds to understand and to remember these things. Prepare our hearts for our good and your glory, we pray in Jesus' name. Amen. Well, I handed you guys, you have sitting in front of you a sheet that's for review for photosynthesis. Remember, you got a test coming up November 5th. Okay? I will have another 1 or 2 review sheets for photosynthesis respiration coming up, but I just wanted to do this one while it was hot from the other day. So one side's completely filled out, the other side has all the blanks. You should fully expect on a test to have to provide all the blanks. Okay? Pretty standard operating procedure around here. That said, we are moving on. We have moved on. We're on DNA. So when I say the genetic basis of inheritance, what do I mean?
DNA and RNA.
DNA and RNA, right? So what does genetic mean? Think about that word. What's at the base of the word genetic?
Gene.
Gene, right? So where do we find genes? DNA. Okay. How many genes are on DNA? 12? What are thoughts? How many genes do you think on your DNA? In one sense, you could say all of them. You don't have any genes that are not on your DNA. In another sense, you'd probably say tens of thousands. Okay. What is a gene? Well, there's that we need the word protein in our definition of a gene, but a protein is not a gene. This is actually the central dogma. Okay. Central dogma is one gene equals one protein. We got to have a little translation in that. We don't literally mean a gene is one protein. What we mean is a gene is the genetic basis for one protein. It's the genetic information for one protein, okay? One gene carries the genetic information for one protein. I've mentioned before, this is generally true. The catch is you actually can get more than one protein out of one gene, but that has nothing to do with the gene itself. It's a term called epigenetics. The word epi means above or on. Epigenetics, so above genetics. So you have little things called SNRPs, SNRPs. And they come along, they're made out of RNA, and they will actually take a protein and they will cut it into pieces. Or they'll take the DNA and cut it into pieces and recombine it in different ways. And so you can get one protein. We have far more proteins than we have genes for proteins. Isn't that fascinating? Which means we have to have some way of doing something with the DNA so that we get all these varieties of proteins, and that comes down to RNA, and it's able to cut and paste different sections of DNA together to get different proteins than you would otherwise. We're slightly ahead of the game there. We started last time looking at the structure and function, the charge and the shape of DNA, because it dictates everything. There's nothing random about DNA. There's nothing unsophisticated about DNA. Now, it's simple. It's amazingly simple. But it's profound, and it's sophisticated, and it's consistent, and it's absolutely beautiful. And if it weren't exactly how it is, life would not be possible at all. We drew some last time. I'm going to hand you guys these sheets real quick. So part of this looks very similar to what we've already done, okay? So you've got two strands, right? Now, the structure of DNA, I've said it over and over again, and do it with me, okay? Please, with your hands, let's do this together. Phosphate, then come down, sugar, nitrogen base. Phosphate, sugar, nitrogen base. Phosphate, sugar, nitrogen base. Phosphate. And when I taught high school, I'd make all the kids, freshmen, seniors, everybody, whatever level class I was teaching, I would make them do a dance. And we would go around and we'd spin, because DNA spins, it's double helix, right? So we go like phosphate, sugar, nitrogen base, phosphate, sugar, nitrogen base, and they get down to their hand. It's a big hit with 14-year-olds. Maybe not so much with you guys, but it's a double helix, right? It spins. The structure is phosphate, sugar. Phosphate, sugar, nitrogen base. Now, it has a backbone. In some ways, people will talk about it being like a twisted ladder. Okay, you ever been on a set of spiral stairs? Right, it's got two handrails, it's got two sides, and then it's got steps. DNA is kind of like that, right? The sides, the rails, what are they made out of? What two things? You should be able to see it in your head, or you could look at it on here. What's on the sides? What's on the rails? And what are the steps? The rungs of the ladder. You got three choices: phosphate, sugar, or nitrogen base. Those are your choices. Two of them make up the rails. One of them makes up the rungs of the ladder.
Phosphate and sugars?
Yeah, phosphates and sugars make up the side rails, right? They're referred to as the backbone, or the phosphate sugar backbone of DNA, okay? You want to make sure that you know that. I need to stop for just a second. I got to address something. The grades in this class are pretty bad. And I've tried to be very gentle and very encouraging and give hope and say, hey, we can, I'll work with you, I'll double test values. But then when I walk around the room and people are drawing, or they're on their phone, or they're generally just paying no attention, not only is that disrespectful to me, it is just really, it does nothing to make somebody want to help you. I mean, you're taking an opportunity, you're taking grace, and you're going, eh. It's got to stop. If we're not going to be serious about being here the rest of the semester, we don't need to be here, because the grades will come out where they've been. If they come out where they've been, then there's literally no point in even coming. So it really is a time to just decide, what are we going to do? Are we here to learn? Are we here to pull the grades out? Are we here to try to succeed? If not, we don't need to be here. Get some more sleep, whatever it is, okay? All right, let's move forward. So we've got a phosphate sugar backbone. Inside you have these rungs. This is completely consistent all the way down. I had you draw this last time, A2T and C3G, and I told you you absolutely needed to memorize it. Just the way it is. There's a lot of information in this, right? It tells me that A and T always go together, okay? If you look on this drawing here, you'll notice that wherever you have an A, it's going to a T. Wherever you have a T, it's going to an A. That never, ever, ever changes. The reason that doesn't change is because of charge and shape. Because A has two rings, T has one ring. When I look at my DNA all the way down it, look, you always see a total of 3 rings. It's 2 rings going to 1 ring, 2 rings going to 1 ring, 2 rings going to 1 ring. But we should be asking ourselves, why doesn't A pair with C? The reason A doesn't pair with C is because of charge and shape. A wants to make 2 hydrogen bonds, and it wants to make them in very specific locations. C wants to make 3 hydrogen bonds, and it wants to make them in 3 slightly different locations than A does. And so, because of charge and shape, A pairs perfectly with T, but not with C. And G pairs perfectly with C, but not with T. We refer to this as base pairing specificity. It's highly specific. To get these things to, I mean, you can put them near each other. They'll fit near each other, but they won't link. They won't hold fast together in a hydrogen bond because the charges and shapes are not in the right places, okay? So, day one, charge and shape, that never, ever, ever goes away. You have to hold fast to that concept, okay? So, when you see this, remember A makes how many hydrogen bonds? 2, with which other base? T. And C makes 3 with G, right? So this is our base pairing specificity. How many rings would I see running across DNA? 3. 2 here, 2 here, 1 here, okay? All right. Let's go back to your drawing here. I want you to look at your drawing. We kind of were here last time. You notice the phosphate sugar backbone on the outside. You notice the base pairing on the inside. C and G are forming 3 bonds. A and T are forming 2 bonds. I don't remember if we got here last time or not. Actually, look at, there's arrows drawn on this, okay? They're very faint on the phosphate sugar backbone. What do you notice about the 2 arrows? They're going in opposite directions. These 2 things run counter to each other. Does that make sense? So when I had you draw it the other day, we did something like this. It went A, T, C, G, and then I sat here looking like a dummy going going C, G, A, T. It's because they literally do that. Okay, they don't go any other way. They have to go contrary to each other. This is how it works. This is extremely important. They have what's called a 5' end and a 3' end. That's referring to the numbering of the carbons on that sugar. I don't necessarily need you to know that, but here's how. Well, I'll give you a second, and then I'll tell you something else. So, DNA is useless if it's not being used. You've got to be able to do something with this. So I've got this DNA, right? It's a long deal. It hydrogen bonds with itself, right? So if I have an A, T, C, G, A, T, G, what should this be? T. And this one? T. And this one? And this one? And this one? And this one? C. This one? T. This one? A. This one? C. Good. Okay. These hydrogen bond with each other, right? One hydrogen bond, not very significant. But when I'm getting an average of 2.5 hydrogen bonds per nucleotide, and I've got thousands or tens of thousands of genes, and each gene has hundreds to thousands of nucleotides, that's a huge number of hydrogen bonds. Think about Velcro. Velcro in itself, a little piece of Velcro, not very strong. But if I had a Velcro wall, and you were in a Velcro bodysuit, and you ran and jumped against the wall, you'd be stuck, right? You could take one little strap of Velcro and just go, rip! But you have a whole thing of it, and all of a sudden you're sticking to the wall. These little tiny hydrogen bonds add up to a big effect. But if all of it's hydrogen bonded together all the time, it's great for keeping it in place, making sure it doesn't get damaged, storing it, etc. But I have DNA so that I can make proteins. I need to go from gene to protein. Your body has to do this all the time. All the time. You gotta make proteins on the fly when you need them, or more of them when you need them, or more of them so that your cells can divide, or more of them so that you can reproduce, or more of them because cells die, and you've got to make proteins, proteins, proteins. If it happens in your body chemically, it's a protein. Anything you do functionally, it's a protein. And you need different ones because they're highly specific. And the way you make proteins is from your DNA, which means I've got to be able to do something with this DNA. I've got to unlock it, okay? So when I do that, I'll take this same strand of DNA, and I'll go something like this. So maybe now I'm going A, T, C, G, A, T, G. And now over here, I'm going T, 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 A, G, C, T, I did that one backwards, A, C, okay? Now, this is open. I can read this. I can do something with this. So, what does your body do with this? I cannot go straight from DNA to protein. That does not work. I can go from DNA to DNA, or I can go from DNA to RNA. I want to focus, we'll show you both. If I were to go from DNA to DNA, in other words, I'm trying to replicate my DNA, I'm trying to copy it. The reason that I would go from DNA to DNA to replicate is because I want my cells are going to divide, and I need to give a copy to both cells. Okay? If you produce a cell and it doesn't have the information to make the proteins, that cell's just going to die right away. It has to get a complete copy of the genetic information to make every single protein that you will ever need. And so what your body does is it replicates the DNA, it makes two copies. One will go to one cell, one will go to the other. Okay? This is called DNA replication. Well, in DNA replication, I'm going to come in and I'm going to go, hey, T, 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 A, G, C, T, A, right? And on the, notice I'm going directionally here. I'm going here, okay? I may actually be drawing this in the reverse order. It's been a long time since I've done this. And on the other strand, these will replicate simultaneously. On the other strand, we would go T, A, G, C, T, A, A, A. So when I say you're fearfully and wonderfully made, I mean, it's amazing. You've got genetic information, tens of thousands of genes that can be turned into hundreds of thousands of proteins, and it's such a simple genetic code. You've got 4 letters, 4 bases that are so highly specific that they only pair with each other to where you can replicate this thing super fast. You can go in both directions. You can do this on multiple points, multiple on your DNA simultaneously. This can zip along, and it doesn't hardly make mistakes because it's so specific. Because A pairs with T, C pairs with G, etc., etc. Remember, how many rings big are these rungs of the ladder?
3.
They're 3. If I make it 2, that means that this thing's going to kink in, right? That's going to be noticeable and fixable. If I try to put 2 and 2, that's going to make it bulge out to 4. Again, that's going to be noticeable and it's going to be fixable. The only time that it maybe wouldn't be noticed and fixed is if I had like an A with a C. That would be 3 rungs wide. But those mistakes so rarely happen because of the charge and shape. Because A wants to make 2 hydrogen bonds and C wants to make 3 hydrogen bonds. It is so simple, it's so elegant, it's nearly flawless. And if it weren't nearly flawless, you would die. Right? So when you get something like cancer, one thing that's happening there is that cells are reproducing just out of control. The reason they're reproducing out of control is something has happened to the DNA. Something has changed. There's been a modification. And now one of the proteins that helps to regulate cell growth is not functioning properly in many cases. And so there's been a mistake to the DNA, which causes a mistake to the protein, which causes the protein not to function the same, and now a cell grows out of control. That's one variant or cause of cancer. You change the DNA, you change the gene, you'll change the protein. If I change the protein, I change its function. You've got to know that, okay? Write that down. If I change the gene, I change the protein. I change the protein, I change the function. Whether that's good or bad, that change, well, it's relative. We talked about sickle cell anemia the other day. Right? In some circumstances, you'd say sickle cell anemia is bad. It can cause pain. It can cause premature death. But if you live in an area with malaria, it's actually an advantage. It all comes down to one, in the case of sickle cell anemia, all that happens there is one of these bases changes. That's it, just one. It changes code, it changes the protein, and on the protein all it does is change one of the 20 of the amino acids. And it becomes a different one, and it doesn't fold the same way, and now at the macro level, the cell collapses. All right. Let's talk about what these A's, T's, C's, and G's are, okay? A equals adenine. T equals thymine. C equals cytosine. And G equals guanine. You need to know that, all right? These are the bases for DNA. Now, I've mentioned several times you cannot go directly from DNA to protein. You can go from DNA to DNA, and you can go from DNA to RNA. You can go from RNA to protein. We talked about this way back when we were doing biomolecules earlier in the year. We talked about 2 words, transcription and translation. Transcription is going from DNA to RNA. Translation is going from RNA to protein. I guess let's throw this one in here, replication. Replication is going from DNA to DNA. Do a chart, compare these two. Let's do DNA to RNA. Start with the D and the R. What is the D? Deoxyribose. And the R? Ribose. So these are sugars, right? When we talk about a phosphate sugar backbone, they both have one, but the sugar is different. They both have phosphate. They both have this phosphate sugar backbone. Yeah, it's the same across them, okay? They both have adenine. This one has thymine. That one does not. This one has uracil. Both have guanine. Both have cytosine. This one codes for thousands or tens of thousands of genes. This one codes for one gene. One more on this chart. This one's double-stranded. This one is single-stranded. Many similarities at the molecular level, the structural level, 2 significant differences. I want you to think about the structural differences that you see listed between RNA and DNA here. What are they?
The double strand, single strand.
Yeah, that comes from one of these other two, though. Uracil is a structural difference. That's a big one. What's the other one?
The sugar that's used?
The sugar. Yeah. So the sugar that's used is part of the reason that, as you were saying, one of them is double-stranded, one of them is single-stranded. Okay? It's an emergent property. So I want to think about this. We said over here A2T and C3G. That has to be different here because I don't have a T. So what are my rules here? A2, U, and C3G. Okay? You got to know that. In RNA, A pairs with U. This would be a classic question on a test. I give you a sequence of base pairs, and I go, what is this? Well, if you see a U, you know that that's RNA. You see a T, you know that that's DNA. You don't really have to go any further than that. If I gave you a sequence, and the sequence was A, T, G, C, A. And I said, all right, give me the complementary DNA sequence. I want to know what's the other, so this is a DNA, okay? Phosphate, sugar. I want to know what the other side of this is. What is it?
G.
Okay? And if I say what's the RNA it's going to code for, what do you tell me?
U.
U,
A.
A, C, G,
U.
U. Okay. They'll look identical, except everywhere you would have had a T, you'll have a U. Now here's our base pairing rules for RNA.
So the
So
T
whenever-
wouldn't be in there.
No T's ever in RNA, right? So this becomes U, A, C.
That would become U too, right? The T would be the T that you already have on there.
No. My DNA does not change. The only question is when I come along to make my RNA, what is it going to put opposite this A? Well, if it were DNA, it would put a T, but it can't. It's RNA, so it has to be a U. The T doesn't change. Let me draw this slightly differently. This will help.
Are you saying you're replicating or you're creating RNA?
Yes. We'd be doing the process of transcription, okay? Let me draw this slightly differently, right? Adenine is how many things big?
2.
2. This one's 1. This is 2. This is 1. This is 2. Okay? So when I come in to put RNA, this has to be 1. This has to be 2. This has to be 1. This has to be 2. This has to be 1, right? And you can see this visually. Now, which one is this going to be? It's going to be U. Which one is this going to be? A. Which one is this going to be? C, and this one? G, and this one? U. That's not a U. Okay. Well, let's practice this. I think this would be a good time to practice. When you were in high school, you would have sheets. I would give you sheets practicing this. You probably have had that experience. And this could go a number of different ways. I could give you some DNA and I go, okay, it's A, C, G, G, C, A, T, T, A, C, G, T. And I'd want two things. I'd want to know the DNA and I'd want to know the RNA. So give me the DNA and give me the RNA.
T, G, C.
Ah, you do it. Don't tell me. So I want to know the DNA and I want to know the RNA. That is not the answer, by the way. That's something else. Got a question, this is a good time to ask it, okay?
last one wrong, because T doesn't follow T, so the bottom one would be A.
Oh, I did mess that up, didn't I? Thank you. I didn't even look at my original one. So some things that we might want to catch. Your complementary, your DNA, and your RNA are going to be very, very similar, right? These are going to be the same, the same, the same, the same. That'll be different. Same, the same, different, the same, the same, the same. The only ones that are different are T's or U's, right? That's it. Everything else is always the same. Because A always pairs with either T or U. T always pairs with A. T pairs with G, G pairs with C. We have the space pairing specificity. Right? Things with 2 rings go with things with 1 ring. Things that make 2 hydrogen bonds go with things that make 2 hydrogen bonds. Things that make 3 hydrogen bonds go with things that make 3 hydrogen bonds. Again, if I had 4 here, it would wonk out and it would be highly noticeable. If I had only 2 here, it would pinch in and it would be highly noticeable. These things would be evident and your body would be able to fix that mistake very quickly. Okay. Another way that I would approach this probably is to give you an RNA strand and go, okay, what was the DNA strand? So what is this DNA strand? What DNA did this come from? Well, U pairs with what?
Oh.
A.
A. A pairs with
T.
T. C pairs with G. U pairs with A. A pairs with T.
C.
Keep going. Yeah, you guys call them out.
T, G, G, C, T, A.
Very good. So we've talked a little bit about the overall for DNA replication and for transcription. And the other step would be translation. So translation, I'm gonna erase over here, okay? You guys have heard, you've written down, hopefully you know that translation is where RNA is read and then protein is produced from it. Okay? Now, before we talk about this, you need to understand there are 3 types of RNA. Okay. First one is mRNA. The M stands for messenger RNA. And this is RNA which will be transcribed into a protein. Or translated into a protein. The second type is tRNA. This is transfer RNA. We'll deal with this a little more coming up. And then the last one is rRNA. This is ribosomal RNA. all have a role in the process of converting the message of DNA into the message of protein, or into the structure of protein. But when we talk about translation, we're talking about this one being read. Okay, this is an exact copy, or an inverted copy, of a genetic message, DNA, convert it to RNA. It's going to be read. It's going to be read by a ribosome. Okay? So the process of translation involves a ribosome. This is just, you need to know, this is the thing, okay? A simple definition of translation is it's a ribosome reading mRNA and producing protein. Transcription, we go from DNA to RNA. Every one base in DNA is turned into one base in RNA. It's a little different when we do translation. There we're going to take 3 bases. Every 3 bases of RNA turns into 1, or is translated into 1 amino acid in a protein. Okay? So these units of 3 are called a codon. Codon is 3 mRNA bases, and they will turn into 1 amino acid. Take a moment to think about something here. Codon is 3 bases, right? Let's do G. 3 bases. Now G can pair with, I'm sorry, that's not right. G can, in a codon of 3 bases, right? I can use combinations of G, C, A, and U, right? What I want to know is just how many different 3-letter combinations can I get with this, okay? So I'm going to be mathematical. I'm going to go G, G, A, G, G, U, G, G, C, and G, 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 okay? And then I'm going to go G, G-A-A, G-A-U, G-A. You don't have to write this down necessarily. Won't hurt you if you do, but you don't have to. G-A, I need G-A-C, and I need G-A-G. Okay? Now I'm going to go G-U-A, G-U-U, G-U-C, G-U-G. And my last one is going to be G-C-A, G-C-U, G-C-C, and G-C-G. So just for the letter G, I've created 16 different codons, right? How many letters do I have?
4.
4. So how many codons do I have? I got 16 just out of G. I can do this for every single letter, right? We did it with just G as the starting one. So if I did it with C, I'd get 16 more. If I did it with U, I'd get 16 more. If I did it with A, I'd get 16 more. 16 plus 16 is 32. 32 plus 32 is?
64.
64. So I get 64 different codons. That makes sense, right? It's cool. 64 codons out of how many bases? 4 bases. Just 4 bases can make 64 different codons. How many amino acids are used to make proteins?
1.
You got to go back to the biomolecule things. We drew proteins and we said, hey, they all look the same except for this R group.
20.
There are 20 different R groups that are used biologically. So 4 bases, 64 codons, 20 amino acids. That means more than one codon has to code for the same amino acid, right? Does that make sense? Only 20 amino acids, 64 different codons. One of them's the start codon, one of them's the stop codon, but that still leaves 62. It means on average 3 different codons will code for the same amino acid. That means that sometimes when I change the DNA, which is called a mutation, that mutation will have no effect whatsoever, because it will still code for the exact same amino acid. And if it does, you'll never notice a difference. But if I make the right change, it'll change the change to the DNA will change the RNA, the change to the RNA will change the codon, the change of the codon will change the amino acid. If I change the amino acid, I may take something that was hydrophobic and replace it with something that's hydrophilic. That's going to change the way, that may change, it may now want to do a hydrogen bond and form a beta pleated sheet or an alpha helix that it didn't want to before. It may change the way that it bonds across it at the tertiary level. It may change the way it interacts with other proteins at the quaternary level. You change the gene, generally speaking, you change the protein. But not always. Some of them, these are called silent mutations. It mutated, you want to write that down. A silent mutation changes the genetic code, but it does not change, it doesn't change anything functionally because 64 codons, only 20 amino acids. Okay? What time is it? Are we at it?
So silent mutation changes the genetic code.
The genetic code. A silent mutation changes the genetic code, but it doesn't affect the protein at all. So this has been kind of a big overview of DNA and RNA and protein replication, transcription, translation. To do more, and we need to do more because you need to know more, it's very hard for me to draw. You need to see, okay? So I'm going to show you some animations.
Over there. Oh. It hurts.
So stretching and it got stuck.
All right,
Yeah.
let's try this one.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add nucleotide, unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded region.
Your DNA is twisted. It is hydrogen bonded together. In order to replicate it, and that's what we're doing here, is DNA replication, if I didn't make that clear. In order to replicate it, you've got to untwist it so that you can access it, so that you can start putting your new bases in there, okay? The molecule that is going to untwist it, and it untwists it all the way down the line, is a molecule called helicase. You must know that, okay? Helicase unzips DNA. I'm not going to make you know these others are called single-strand binding proteins. Basically, the helicase unzips it, untwists it. It wants to just go right back together. It naturally wants to have this twist. Okay? So you have to put something else there to like hold it apart. You ever have a twisted rope and you try to untwist it? As soon as you let go, what does it do? Just twist back, right? So if you had to work with a long section, you'd have to have somebody hold it open. That's what these do. They're holding it open, okay? So that replication can happen. So far, the enzyme you must know is helicase.
Of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA.
Helicase unzips, primase. You have to know these. Helicase unzips the DNA, primase starts replication with RNA primers. So for a minute there, we'll have U's in our DNA, right? But those are going to get, and we'll have ribose in our DNA. That will later get taken out. But to start off, you have to have it. So helicase, primase, and then polymerase. If I could spell it, it'd be great. Polymerase is going to add DNA nucleotides. start.
Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
Let me show you what that was trying to show you. Okay. So my DNA, I open it up, right? Who unzips it?
Helicase.
Helicase. Things happen directionally. This has a directionality. This has a directionality. As this way, this would be what I call my leading strand. Okay, so polymerase can work continuously in this direction. As this unzips, it can just keep building, boom, 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 boom. But because it's directional, it cannot do that down here, because it's anti-parallel. Down here, it's got to go this way. Well, since I don't have this, if I had this just totally unzipped all the way, then yeah, I could just start down here and move here, right? But I don't. I'm going bit by bit. So it's going bit by bit. It's doing it in fragments called Okazaki fragments. Now, we can work both directions on DNA. So down here on this end, I can go continuously. Once it starts, I can just run forever in this direction. But on this end, I can't. Wherever it started, I can go, and then it's unzipped, I can go, and then as it unzips some more, I can go, and when it unzips later, I'll go, and it'll make Okazaki fragments. So on each of the two strands, you have a leading end, and you have a lagging end. This is leading, this is lagging. This is lagging, this is leading. Okay? These little things here are called Okazaki fragments, named after the guy who found them. Remember, polymerase cannot just start making DNA out of thin air. It has to have an RNA primer in place in the first place. So I will have an RNA primer at each of these sections. That's going to have to be removed. You lay down a primer, and then polymerase can do its job. You lay down a primer, and then polymerase can do its job. You do the same thing over and over and over again. This is how it works. So these red or these pink sections are RNA primers. You lay down the RNA primer, and then polymerase can go. It can go all the way in on the leading end as this unzips. On the lagging end, we can go as far as it's unzipped, and we work backwards. Then when it unzips some more, we'll go there and we'll go backwards. It's constantly doing this. One side will be going this way, one will be going backwards all the way. That is somewhat inefficient, because I'm spending a lot of time making RNA primers and a lot of time making Okazaki fragments that later I'm going to have to come and replace that and fix that. But this is how the Lord engineered it. This is how the Lord engineered everything. Every living cell of all type is like this. Okay? So, at this stage, we've got chunks of DNA, chunks of RNA, or long strand of DNA, chunks of RNA, chunks of DNA, chunks of RNA, chunks of DNA. Opposite sides, he used the word anti-parallel. That's a really useful word. They're parallel in that they're going, they're running side by side, but they're going in opposite directions, so anti-parallel. I'm going to run this whole video one more time. We're going to catch a little more out of it.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bond.
Stop there. This is the last. I don't need you to know the difference between polymerase I and polymerase III. You just know they both lay down DNA. You do need to know that ligase is the one that will remove the RNA, or I'm sorry, ligase is the one that will attach the phospholipid, the phosphate sugar backbone. Okay, so we're going to add that one. Ligase fuses the phosphate sugar backbone. What is this? What process is this?
Replication.
DNA replication. What are the pink things?
Primers.
Yeah, RNA primers. They need RNA primers because polymerase can't just start laying DNA out of thin air. It has to have a primer in place. That creates a problem because I'm going to have chunks of DNA and chunks of RNA. So another polymerase is going to come and remove that RNA and replace it with DNA. Who's going to fuse the little segments of DNA together?
The fragments.
Yeah, what's going to fuse all that together? It's up there.
Ligase.
Ligase. Good. And what are those fragments called?
Ozarki.
Okazaki fragments. Close enough. Okazaki fragments. Shish kebabs. Okay. And why do they form? Yes, but one side doesn't have them and one side does. Why does one side have them and one side doesn't? One side's lagging. So as it can only work as it opens up. It always is working in the opposite direction. One can move into the opening, and one has to work away from the opening. So it creates these fragments, right? Okay. There's a lot in a simple 2-minute video, isn't there? Okay. Well, I'll put, obviously the whole class video will be up, and I'll also link this video, okay? So it'll be there. I'd watch it, and watch it, and watch it, and watch it until, like, I could do it with the sound off and I could narrate it. That would be my goal. You ever seen a kid, like, they can't read, but they can read a book because they've had the book read so many times, they can just tell you, yeah, that needs to be all. Okay. Just tell me. How long would that take? I mean, if you did that 4 or 5 times, you'd have it, right?
I don't know about that.
6, 7, 8.
Yeah.
Literally, we're at 1 minute and 45 and we're done with this. You know what I mean? It's
It's
not long.
As of this song, there's other reading.
Okay, well, let's take on RNA.
She's so funny.
Okay.
Thanks for stopping by. This is 2 Minute Class, and today we're talking about RNA, specifically mRNA, rRNA, and tRNA. Let's start with mRNA, or messenger RNA. So named because it acts as the messenger between DNA and protein production. And if you want to know the difference between RNA and DNA, I have a great video for that linked here and in the description. When you discuss RNA in school, you're usually learning about mRNA. During transcription, DNA is transcribed into mRNA. This mRNA is then translated into protein. It serves as the genetic template of a protein since the DNA can't leave the nucleus. Next we have tRNA, or transfer RNA. This is my personal favorite, and honestly one of the most fascinating molecules in genetics. RNA is generally single-stranded, but tRNA actually folds back onto itself to form a distinct T-like shape. Without going into too deep, transfer RNA is responsible for bringing amino acids together during translation to form the growing peptide chain that will become a protein. And last but not least, we have rRNA, or ribosomal RNA. This RNA is the main component of ribosomes, which I've discussed in a previous video. rRNA combines with special proteins to form ribosomes, which then read mRNA to form proteins. There's a lot in that sentence, so feel free to jump back and listen again if you need to. But essentially, ribosomes contain rRNA and help build proteins. To bring it all together, sections of DNA are transcribed into mRNA, which is brought from the nucleus to the ribosome, composed of rRNA and protein. The mRNA is then read by the ribosome, which brings the tRNA in with the appropriate amino acids to build whatever protein the mRNA is a template for. Now watch these related videos and hit that like button. And let's see how quickly we can get 100 likes on this video. Thanks for watching, and we'll catch you next.
They say no.
Give them a like.
Go away. Okay. All right.
Alright, for us.
In order for our bodies to function
No, start.
with a variety of nutrients we get from our diet, our bodies cannot use as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into useful nutrients our cells absorb. And the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template
Look like a
DNA
monster eating
string.
you.
As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns.
We're going to stop there. So, transcription is the process that we were just looking at. Transcription is going from what to what? DNA to RNA. What is the only enzyme you saw involved in that? RNA what?
Messenger.
No. Polymerase. So the only enzyme you need to know for, you had a bunch for DNA replication, but you have one for transcription, and it's RNA polymerase. You've seen the word polymerase before. It was there with DNA. Polymerase added DNA nucleotides one at a time. RNA polymerase does the same thing, but in RNA. That's it. I will see you Tuesday? Today, Thursday? Oh. All right. God bless y'all. Have a good day.